So today I'm going to talk about a biology-driven framework to guide cancer therapy. Two disclosures: I have a company Unravel Genomics, not discussing any of our biomarkers, and then I also work at ARPA-H, U.S. government. I am not representing the government today. And the work I'm presenting is from my work at City of Hope, and a lot of it has been driven by my mathematician colleague, Jason Griffiths, shown there, who is just brilliant. And I know he would have loved to be here, so maybe next year I can direct him to this meeting. Okay, so just a little bit of background on what I'll be talking about today is estrogen receptor, or ER, positive breast cancer. Breast cancer is similar to all the other cancers we've been describing today, where we can have a heterogeneous tumor, and that's kind of represented here, where the purple cells are kind of different clonal populations. The other colored cells are other normal cells in the tumor microenvironment. And a tumor can start out sensitive, but evolve during therapy to a resistant state, and that is represented by those darker purple colored cells. And so in breast cancer, the majority of patients are estrogen receptor positive, so there's a large population of patients. And for these patients, they're typically given therapy called endocrine therapy that targets estrogen signaling and cell cycle therapy. This is standard of care for these patients. Now, if you're stage 2 or 3 ER positive breast cancer, you have a 40 to 50% chance of recurring with metastatic disease. And if you have metastatic disease, all those patients, or almost all those patients, progress to refractory state, where we don't know which therapy will work best for them. So it's really important to be able to understand the dynamics of cancer and to be able to predict a tumor's trajectory, and not just that, but the sequence of therapies that would be most effective at a durable response for these patients. So that's what I'll talk about today, a framework to get to that point. And really the goal is to predict high-risk patients or for stage 2, 3, so we can give them additional therapies that prevent that recurrence from happening, or for metastatic patients to optimize the combinations and sequences. Okay, so today I'll just be talking about data from a clinical trial that I was happy to work with others on, called Feline. It's actually in humans, not cats, but I don't know why they called it Feline. And this is for ER-positive, high-risk stage 2, 3 breast cancer patients. And in this trial, it was a really controlled setting where patients would be enrolled on day 0, and they'd get a biopsy. They would then, at day 14, get a repeat biopsy, and then at end of treatment, after 6 months, they'd again get another biopsy. At the same time, we're getting biopsies for molecular data, which I'll be talking about today, single-cell RNA sequencing data. They're also getting clinical measurements, some of those shown down below, to understand if a tumor is growing or responding to during therapy. And just to kind of note the treatments, letrozole is an endocrine therapy, and then letrozole plus ribociclib is endocrine therapy plus cell cycle. So the clinical objectives of the trial were to understand whether or not patients in this type of disease state respond better to combination or single agent therapy. That's not really what our question is. They kind of target the same pathway, so we wanted to understand how patients progress on these therapies. Okay. So again, for these patients, they're potentially curable, right? 50% of these patients will progress to metastatic state. We don't know who they are, but if we understand the biology, we can perhaps cure some of those patients. Okay, so the objectives of our study were to really build a long-term framework where we take discovery from patient data. Then whatever we discover, because we're very cautious, we want to test these findings in the lab, so I'll show a little bit of in vitro work. And then the long-term goal, right, is to deploy whatever models we're building into patient care, so that if you are a breast cancer patient being seen in the clinic, what we've learned here will benefit you. The research questions, biology-based questions we have, so I realized I didn't put the math questions on here, but they'll come up, are really, what are the reproducible resistant traits in ER-positive tumors as they are treated with these standard of care agents? And if we identify these resistant traits, how do we then test what treatments targeting these states, how they will work? So that's the in vitro work. And then lastly, it'll get, I'll throw in the math, so I like the inverse direction of some of the earlier talks where we try to make forecast drug response and make predictions on how a patient will respond to treatment.
So may I ask you quickly, do all these patients in the trial receive the same treatment?
No, there are 3 arms, right?
There's 3 arms.
Oh, sorry, I missed
All
that.
patients receive endocrine therapy. Some patients receive cell cycle therapy also, and there's different dosing timing. The different timing didn't end up being different for the patients. So you can think of it in your head as like endocrine therapy for everyone plus cell cycle for some.
Thank you.
Are they randomized or is it some other
They are
criteria?
randomized, yes. Okay, so there were a total of 120 patients of the cohort. Of those, we got good data from about 60 patients, meaning we got single-cell RNA sequencing and clinical data at all 3 time points for 2 different cohorts. So we split the trial into earlier patients, 35 patients with 3 time points, and luckily about half are responsive to treatment. And then we have a validation cohort of 27 patients. From these biopsies, we performed single-cell sequencing and DNA sequencing on all 3 time points, and we have over 400,000 cells, which are underlying the analysis that I'll show you. Yes?
Sorry, because it might, sorry to interrupt, but because of tumor heterogeneity, are you biopsying from just a single site in each patient, or do you have multiple?
Yeah, so unfortunately, so it's neoadjuvant, which means that they don't, so the patients enrolled, we only get a biopsy at day 0 and 14, and then at 6 months post-treatment, we get the whole tumor. So we do surgery to remove the tumor. And so for those first 2 biopsies, it's just a single needle, single
biopsy,
place. Yes.
Oh.
But there's still a large collection of cells.
I don't know
Yes.
how many. Is that
Yes.
large enough?
Yeah, it's a pretty big size biopsy. In order to get single-cell RNA sequencing, you need a fair amount of material. So, and even though I'm talking about 400,000 cells, there were more that just didn't pass our QC, right?
And there's not, I don't know anything about this, is there not that you might have very different clones in different parts?
You might have different clones in different locations. However, we have some power in the fact that we're looking within a patient over time. Most times you just have a upfront time point, right, across diverse patients. So your power to identify differences in responders, non-responders is based upon pulling that information from a very heterogeneous population. What we're doing is we're looking across time, where we have one patient, even though the biopsy might not be in the same place, it's still a lot more powerful.
Okay, thank you.
Right, basically, the assumption of a biopsy, more or less, is that it's a well-mixed population, even though it probably isn't.
Probably not.
No, it probably isn't.
Okay,
Yeah.
okay, thank you,
So,
thank you, I'm sorry.
Yeah, I'm always greedy, but pathology won't give us all they take more biopsies, but they're for clinical measurements. Yeah. And there are other tests being performed, like Ki-67 is part of the trial. So, okay. Now, this isn't a bioinformatic talk, but I just wanted to reflect, there's a lot of bioinformatic analysis, some which Ben talked about earlier, where we identify subclone populations, mutations, copy number variants. I'm going to just briefly touch on the DNA. And then what I think was really fun for us is to work with our mathematician collaborators to understand things like phylogeny across the single cell populations over time. So there's a dynamical component. How to classify, you think immune cells are one population, but it's thousands of populations. So how do we build better classifiers? How to look at cell communication across populations, and then some predictive models that I'll show later on. So we're always looking for collaborators. I just wanted to throw that in there. So if any of these questions are interesting to you, just feel free to reach out to me. So the first question we had is, how do we take so in ER-positive breast cancer, the tumors grow pretty slowly. And the radiological and other measurement modalities tend to be very noisy, and you don't oftentimes have frequent measurements. And so you have to build a model that represents the diversity of data and then build these projections out over time so we can understand whether a patient's responding or not. It's not so easy to classify stage 2, 3 breast cancers as responding or not to therapy. So the first thing we did was we took all the different measurement modalities that these patients had, including physical examination where they measure the tumor with a caliper. We took the mammograms, the MRI, the surgical pathology, which is the ground truth at end of treatment. So that's that blue dot in. The pathologist tells us exactly how big the tumor was at that point, so that's true. And ultrasound. And on the top right, you'll see like these measurements have different features, like they can be noisy or biased. And our model accounted for these different limitations of the data to build these trajectories across the 120 patients so we could classify them into different groups. So the model used for this was the dynamic Gaussian process LVM, and that was really just to capture these nonlinear relationships between the different modalities. So as we looked at the data, we saw that there were some responsive patients, you see in the left in the red and yellow, that had either a sustained or partial response. And what you're looking at here is like the tumor trajectories from day 0 to end of 6 months and how the tumor growth rates were over time. You can see these patients were pretty much responding to therapy, whereas those with stable, rebound, and progressive disease were, we call these growing.
These pictures are not from experiments. They're derived.
They're modeled off of the actual data. Yeah, these are just representative patients where you can see the different measurements at different time points.
For the same patient,
For the
you
same
have
patient, right.
every 2
Yes.
weeks or so you have
Yes,
one
so
of
each
those.
patient had a plot that looks like this, and then this shows all the patients that followed that type of trajectory. Yeah.
So maybe I'm misreading on the colors, right? So those are red dots that you're showing, right? So that's physically,
Right, so the physical exam was when the patient went into the clinic, and the clinician took what's called a caliper and like,
okay. But the mammogram doesn't happen every time, MRI doesn't happen every time.
exactly.
Okay,
It's very
got it.
sparse data that's sporadic, doesn't fit any time frame other than like the surgical pathology is always at 6 months.
Okay, got it. So
So-
we can't think of this as a 6-variable system.
No,
Got it.
yeah.
And the physical examination numbers are terrible, probably.
Yes.
So how
Yes.
much,
And-
in doing this computational biology, how much trust do you put on that?
Right, so you can see that the model then accounted for the
I was looking
precision,
up there.
right?
yeah,
So
yeah.
it learned that those measurements were not precise and variable based on the doctor doing the measurement, right? And probably the location of the tumor.
I'm sorry, just to confirm, so in the last graph, so these multiple curves that you're doing, the colors actually do not correspond to the...
Sorry,
No.
these dots respond up here, and this is just like
Different graphs.
each line is a patient. Sorry, and they're
The
just
blue there is a different blue, it's unrelated to
unrelated,
to the
sorry.
pathology.
Yes. My mathematician just likes pretty colors.
But the same pretty colors, that's for sure.
Yeah.
There are more colors.
Oh, yeah.
Is the blue shading your?
Oh,
like,
that's the uncertainty in the predictions
How
of
is
the
that
trajectories,
calculated?
yeah.
How is that calculated? Off your trajectories underneath?
That's calculated from the different data measurements.
That's the variation in the Gaussian.
Yes.
Which Gaussian?
Yeah, which
This is a
Gaussian.
Gaussian.
This
Oh, okay, okay,
GPLDM.
okay.
But I thought that was not model.
But the top then is from the model, but it also has the data overlaid.
Yeah. I see.
Sorry, the top then is from the
The
data.
top curve,
Right.
the black line is your sort of
Is-
average,
It's the
Yes,
model.
and
yes.
then the data is the dots laid
Yes,
over top of
yes,
the
that
model.
the uncertainty is the blue.
Okay, thank you.
What's the difference between a partial response and a sustained response? Because to me, it looks like the partial response is maybe a slower response, but still going down.
The partial versus stable.
No, this partial looks like it's just declining slower.
Oh, sustained. Yeah, in general, it's based upon the slope, the slope severity. If you have a more kind of like a stronger response initially and it stays down, that's more sustained versus if you have this more gradual kind of response over time.
But the gradual
That sometimes, and you can see sometimes these actually kind of go back up like that, whereas these are all, like if you see where they end up, this kind of ends up here where these all stay
Right.
really low.
So how was this classification done? By a doctor or by?
This is the mathematician. Yeah.
Well, the y-axis on the upper left sustained response and the upper right leftish partial response, the y-axis is totally different. So the partial response, the slope is much, much less than the slope on the
Right.
sustained response, which is what you were saying.
Right.
It's based on slope.
I want to get to the fun part, so I'm going to keep.
Sorry.
That's funny. We all, I got to talk faster. Here are the single cells. Here's the classifier. different colors represent different cell types. The pink ones are the cancer cells, and this is just the distribution of the different cell populations. And Ben talked about these subclonal populations. Everyone a lot of cancer biologists think that much of the resistant state is driven by genetic mutations. And so in these patients, we performed from the DNA now. So we're flipping back to DNA. We're pulling out the copy number alterations, the DNA mutations. And based on that, using the methods that Ben talked about 2 days ago, we plotted out the subclonal populations. These are what's called fish plots, and the different colors just represent different cells within a tumor that have different genetic mutations. So they're considered different subclonal populations. And then this is how they evolved over the 6 months in the initial cohort. Yes.
But it's just based on these 3 temporal measurements, yeah? So like day 0, 14, and 180.
Yes.
There's none in between, so the rest is just...
That's right. So those are just they're not even modeled. It's just the fish that shows the whole thing. Yeah. So this was just actually 3 time points, and then okay. But on the right here, you see some of the common driver mutations in these patients. And I'll just give you the conclusion, which was there weren't any key driver mutations that were common across our patient population. So we knew that we had to put more weight on the RNA sequencing data. So to do this, we took, again, the 3 time points, and we took the cells from each time point and performed what we call SSGSEA. It's just a way to not look at individual genes, like MYC, for example, just one gene's expression. You take a pathway of genes, like 150 genes that are in the MYC pathway, and then you summarize their score. And this is just a more robust way to look at the biology of a tumor because there's high variability with individual genes. So we perform.
It's like you could take an average, but you don't take an average. You try to do something that's more.
Yes, it's a little more sophisticated, but
a little more appropriate for the same.
But it's kind of boring, so I'm not going to talk about it. But so you get this pathway score across these 3 time points, and then for us, we needed to understand the dynamics of the different pathways. So we would take the key oncogenic pathways that have been discovered through history, there's large databases that summarizes these pathways, and we performed a hierarchical regression model across the 3 time points to identify those pathways that were most dysregulated. And the top pathway we saw in these patients was really a change in estrogen response signaling. So here what you see is the hatch lines represent individual patients, where the solid line is the average across all patients for the regression modeling. And red represents tumors that were classified as growing or resistant, and the blue represents patients who were, had the sustained response or, response to the therapy. And so the first thing we saw was that the resistant patients, this isn't at scale, this is the 2 weeks and this is the 6 months. The first thing we saw is that even within 2 weeks, we saw a change in estrogen signaling, where patients who responded to endocrine therapy, their estrogen signaling went down, right? So that's what you hope to see in a patient. That's what you want to happen. But for those patients who were classified as growing or resistant to treatment, their tumors continued to have estrogen signaling, and that signaling didn't decrease. And that was with single agent endocrine therapy. The other shocking thing we saw was that for combination therapy, you again see that responsive patients, their signaling goes down. But for resistant patients, because you're hitting 2 nodes of this pathway, right? So estrogen, cell cycle are within the same kind of signaling pathway. We saw an independence from estrogen signaling. They're rapidly developing estrogen signaling dependence, and they're becoming more what we call basal-like, which is a different breast cancer characteristic. So, and this is just another estrogen signaling pathway. So we saw that there was this strong kind of decrease in estrogen signaling, and we wanted to understand what are the compensatory pathways that are driving proliferation. So we also looked across not just estrogen signaling, but there were other pathways that were modeled in this system. So this is the same analysis. I'm just showing a different pathway. This is what we call ERB pathway. This is a growth factor receptor pathway. It's just a very popular signaling that can drive proliferation independent of classical cell cycle and estrogen signaling. And this was another top hit, which where we saw in resistant pathways, there was a significant ERB pathway activation. So this is patients pre-treatment and post-treatment. So yellow is pre, red is post-treatment. You can see in approximately 2/3 of patients in the discovery and validation cohort that the ERB signaling would go up significantly. So this was our first hint that this was a compensatory pathway based upon the modeling of the dynamic data from the feline patients. Now, we like to kind of confirm results in other settings before we take it further, because you can always get like spurious results in data. So we also can currently test things in the lab. So I have a lab at City of Hope, and we developed this system where we grow cancer cells, so ER-positive breast cancer cell lines. We grow them in 3-dimensional culture, and we kind of grow them for up to 21 days. We can stop the experiment at any time, of course, and we can treat them with therapies and look at how the cells grow. So we can also do things like Western blots, which measure protein expression, or RNA-seq, or any other types of assays from this data. And I don't want to bore you to tears, so I know this is like, no? Okay. This is like experimental data, but it just shows in 3 different cell lines that have different lineages. We show that when we treat with endocrine therapy, which is the red, compared to control DMSO, we see for each of the 4 ERB receptor family members, we see they're significantly upregulated after just 7 days of treatment. So what we're seeing here is a rapid upregulation of ERB upon endocrine therapy resistance that tracks with the rapid upregulation of ERB signaling in our patients. So that was kind of reassuring. It's a simple assay, but
Sorry, these cells are, first of all, do they ever reach confluence in that time and you have to replate them, or do you?
Yeah, so this is what it looks like.
Okay.
Okay, I will flag here that my mathematician hates this. He goes, it looks like a ball, so he just hates looking at this. So I'm always hesitant to show it, but you like it? Okay, I will tell him. So this is what's called a spheroid. It's like a ball of cells growing in the little well. So you have media in the well, and the ball of cells grow. And they're labeled. They have like just a GFP label or RFP label, so you can actually visualize them. And then you can also measure their growth, so you can make it into an actual like measurement, right? And it's all high throughput. So these are cells that have grown for 21 days in the 3D spheroid. And you can see that with DMSO, they're growing great. Both populations are growing great. The cells that are both sensitive to endocrine therapy and resistant to endocrine therapy, because they're just growing in normal media. And then if you add fulvestrant, the sensitive cells don't grow. They're small. There's less of them. The resistant continue to grow. If you add an ERB inhibitor called afatinib, it's not important, you can see that both cells actually respond to afatinib. And if you combine afatinib with the endocrine therapy, you get much better response than just fulvestrant alone in this resistant population. And this is just quantified here, so you can see the differences. So we get measurements through all the time points. So we make measurements every couple of days, and we have other work with these types of populations. So if you're interested in modeling kind of cell dynamics, you can look at these papers. Happy to share data. Okay. So far now from both the patient data and the experimental data, we have this new kind of compensatory pathway, or B, that can lead to ERK activation and estrogen and turn back on kind of estrogen-related signaling pathways and cell cycle proliferation. This was seen in both the discovery and validation cohorts and the experimental cell lines, and we see evidence of biological evidence that this is a kind of common mechanism where ERB receptors are upregulated at the protein level. So it's not just RNA level; we see it at the protein level from the experimental data. We didn't measure protein in the patients because we don't have that data, but and then we can see that ERB inhibitors, I just showed one experiment, but there's a lot, that resistant cells can have sensitivity to these ERB inhibitors. So the next thing we want to do is try to see if this can be a therapy for actual patients. In order to do that, you would need to, a patient would come through the door, you need to be able to project which of the patients would have this as an acquired resistant trait. So that's kind of the next question we asked. And I kind of just reflected that here. So current state is, if you're a patient, you walk in the door, you might get a measurement up front that says, okay, these pink cells are, estrogen receptor positive. So you give endocrine therapy. You always had the resistant cells there, but because we don't take measurements a lot of times, you don't know they exist. Tumor is still shrinking, so you continue to give endocrine therapy, but tumor is still evolving, so you get these resistant cells. Now they're light blue and dark blue. You switch therapy, but they're not directed to that particular resistant trait, and the tumor continues to grow. So ideally what we want for our models is to predict that these cells exist, add on this therapy, and then continue to model the disease trajectory so we can continue to give the right sequence of drugs. That's kind of just the model kind of goals. So to do this, we've started building out a process to kind of project out what those states will be over time. And in order to start this process, we built, Jason built a synthetic data set. Oh, is there a question? Sorry.
I don't want to derail you, but are you focused on cancers where there's not too much of an immune component and so the organoid model makes sense, or how do you feel about that part missing? Just honest question, not trying to.
Not a problem.
Yeah.
We see two main signals. I didn't talk about the other one.
Yeah.
So in the combination therapy, and we just had a paper on this in, I think it's Nature Communication. In the combination therapy, not endocrine therapy alone, we also see dysregulation of immune signaling, particularly interleukin-15 signaling. So in that context, we're doing multicellular 3D co-culture models to integrate the immune cells with the cancer cells and then look at why does the CDK inhibitors, why is there an immune component at all? What do they do on the immune cells? How does it impact cancer cell growth? We have that data as well that's published, so you can look it up for sake of simplicity and just giving the overarching structure of our pathways. Yeah, but it's a great question. You'll see it reflected actually in a couple slides that I have too. It's a great question. Yeah. But it doesn't seem to matter for endocrine therapy, so it's kind of an interesting.
Right. It seems like that could be a good model then for the organoids.
Yeah, but for combination therapy it matters. So yeah, like in order to get to the predictive state, we built the synthetic data, and we have to account for different challenges, right? So we have patients over time, over phenotypes. The data in the single cell or other kind of data we're collecting on patients in clinical trials is sparse and noisy. And in order to kind of model this data, Jason built out an adaptive dynamic ODE to simulate phenotypic coevolution. So right in line with your question, like oftentimes we'll see like growth factors go up, immune signaling goes down. So we'll see this kind of traits reflected, like complex traits reflected in the data. And then the real goal here is to build a predictive model that accounts for these features and can project out where a tumor is going to go, what are the resistant traits that will drive their progression.
And you're going to show us the equations of how the immune system has been affected by the.
Oh, you want the immune system? I don't have the immune.
I'm curious to know, because we did a lot of models of this, and I just want to understand how you're modeling the effect of the immune system, because it could be immunogenic. It could be that it attracts immune system.
Right.
It could be strong.
Right. So this model isn't driven by any one biological. So this model doesn't have priors for the biological traits. The representations from the data, like the different features that represent different biological states, but they're not predetermined, if that makes sense.
No, what I'm saying is, maybe I misunderstood, but in your model where you'll have components for various parts of the immune system, T cells, B cells,
No, but
macrophages.
she's not doing that. They're just going to be learned from the data. They're going
I'm
to be
sorry.
inferred.
Oh,
I
I
didn't
thought you said
present that paper
I thought you
today.
said synthetic data.
I do have a really good paper on that, but it's not part of this slide deck.
Okay, because it says synthetic data, so I thought
This
it
is
wasn't.
synthetic data just to
So
look
it's generated
like the real
in some
data,
way.
so that we can generate where I'm going is we need to generate a GPVAE to learn the tumor dynamics and make predictions over time. So this is how we're building these predictive models, so that when a patient walks through the door, we take their data and then we can understand the tumor trajectory. So we're pivoting from understanding biology in a tumor to making a predictive model. Does that make sense?
I just don't understand what it means.
She's not doing
data.
modeling in the
But
sense
what
that
is
you're,
synthetic data?
It's not-
it's generated from previous data and you're doing.
Oh, I see. It's just like bootstrapping from previous data
Sorta,
or
it's not-
something.
it's
Oh,
not-
I see.
But I
Okay,
think-
sorry.
I
Sorry.
think
Got
also
it,
those
got it.
trajectories, if I'm understanding right, they're learned from like some kind of latent space embedding, right? So
That's right.
it's like you might learn a B cell component or trajectory, but it would be inferred from the data. It's not because you
Right.
know a priori that those are B cells that are included in a model, not like an agent-based model or something like that. It's just that it's inferred because they move differently, like immune cells. That would be
Based on the experimental data.
Right.
based on the experimental
yeah.
data. So it's data-driven.
Synthetic in the sense of bootstrapping or
But like
something.
that
not-
synthetic data was just a test, right? Here you're using real data, so.
Right, my apologies, I pivoted a little fast. In a separate line of inquiry, we have the single cells, and we have their cell subtype population characterized, and we look at interactions between those populations using the modeling that you're referring to. And that's in the paper that I didn't discuss today. But this is about building on what we have to impact patient care. So my goal is always to try to take what we have and get it to the clinic. So I need some model that I can use to take a patient sample up front and predict, no, that's fine, it's my problem, and predict what's going to happen in the future, right? So that's what this is. So for this type of approach, what Jason has taken, and I want to take credit for his work, is this variational autoencoder to learn these latent tumor dynamics based upon those phenotypes I told you about from the single cell data. So here we're going to take the same patient data, the pathways over time across the different cells, and then learn those to make predictions. So as part of the VAE, we'll have this complex data, meaning all these different phenotypes, right, that we've classified from the gene expression data. And that will then go into this latent space where we, these little lines represent different patients, where we need to know, like, if these are the pinnacle of resistance to the refractory state, we need to know where the patient is on this landscape so we can treat them with the right targeting drug that targets the resistant state. So that's the goal. So we take the data, we have these latent states, and then we understand what they are at the back end. So some of the key features of this, the innovative part of this really is the GP prior over the latent trajectories. And what that does is allow us to have this component of time and coevolution of phenotypes so that we can make these projections. Yes?
You're able to get to this latent, to good representation of this latent space just on these 35 patient trajectories.
Yeah. Well, I'll show you, and you can tell me how good it is.
You know better than I do.
Yeah, I mean, I think it's reasonable. I believe that I did launch a national clinical trial where we'll have 1,700 patients. So I think that will be much more powerful than, but where we have strength here is really building again from those sequential biopsies. And then we already do dimension reduction on the gene expression into pathways. So we have some type of,
Like, it kind of comes down to the question that you say, okay, the patient that looks similar on your sequencing landscape also behaves similar.
yes,
In like over time.
right.
I mean, the one, the UMAP thing that you showed, I think this was an overlay over all patients, huh?
That's all patients, yeah, and
So
all the
that's
different
also
cell
the
types.
question also how, this cancer
Right.
population was large, how individual patients show up there, and they're probably not all in the same spot, so.
Right. That's a great point.
Is it
Yeah.
feasible in the sense that you see, okay, if this is the UMAP picture of a certain patient, it also has a trajectory that is similar to, there should be some
Right.
similarity between the UMAP representation somehow and the trajectory. Is that?
Yeah, I didn't look at that, so that's a great question. Does the UMAP classification have any bearing on,
At least this is what you assume here. Huh?
Yeah.
I mean, this is what you
Right,
kind of.
right. But what we want to do is learn, so let's say we have 1,700 patients going through multiple lines of treatment. We want to learn from those dynamics, what the next patient looks like. So the UMAP reflects just the classification of where they are in space, but I feel like there's a dynamical component we need to kind of, okay. So we first applied this VAE to the test data that we had, synthetic data, and just to see how the model is performing. And I kind of misordered these. So on the right is just the comparison between the reconstruction of the data and the input data. And as part of the model, there are 12 different dimensions in that latent space over 3 time points. And I think this is just the synthetic data, I think was around 15 patients. You can see that there was a strong correlation between the mean reconstruction across the featured dimensions, which shows that they are preserving some of the biological meaning of the data. And then on the left is the ground truth from the synthetic data is the top. And again, each line represents a different patient in the direction that their tumor is going in across those different axes. So shown here is just axes 1 and 2. But then you have the different patients. And you can see that there's a pretty good mapping, so this gets to your question, of the patients from the learned latent trajectories compared to the ground truth. So we felt good that the directionality, the speed of recovery, and the parameter accuracy were pretty good here. So we moved forward to the actual data. This is based, again, on the feline data. This was synthetic data. And that's shown here. So for this analysis, again, we took the feline data across the 3 time points, and that's each patient is a different color, and and this just shows the direction of the latent feature. And this analysis, I just gotta say, did not include like ERBI as a target or prior results from or patient outcomes. So it just shows kind of the representation of the data. So you can see here that a lot of the patients are going to the right. They're going right here, and this is the first latent space of the model. And when we looked at the feature, like the correlation of the feature to the biological pathways that are part of this data, those pathways I already told you about, those are shown here. What we see is that the EGF pathway in multiple settings, these just reflect EGF, and this is a downstream target of EGF, was found highly correlated to this latent feature 1, which is what's here. So at least for the feline patients, this is the top feature that depicts the growth of these, direction of these samples. So it seems to be predicting biology. The other feature was immune signaling. So that may or may, like immune signaling is complex. I'm not saying this is the same immune signaling that we saw in our other paper. We need to dive deeper into it, but it does reflect that immune signaling is also important for these patients, at least a subpopulation of them. So, so the result is that these predictions seem to be working reasonably well at this point in time. Yeah.
This is like representation of your latent landscape, so
Yes.
to say. And there are some parts that are like densely sampled, and some they are like really sparse. I mean, it's like that you kind of get an idea what happens there in the middle. I see the point, but for these others, it's you don't really have many representations there.
Yeah, you know, I think, so this is what, 50-some patients. I think as we get, I think we do need more, like we do need the 1,700 patients to really find, like if 2/3 of patients go through this acquired or B signaling state, and even some of those that don't might still have some component of it. It might be, of course, multifaceted. You know, as we get more patients, we'll see more divergent and more trajectories that overlap across those other groups. And it could be that we're just not showing, like this is the second latent feature, but it could be that the 3rd or 4th really characterize these other people. Yeah. Still, I'm happy to hear feedback on how we can improve or other methods to like make predictions, because we are moving forward with like we'll get a lot of clinical samples and we'll need to make these predictions. So this is just kind of one solution. There could be others. I'm sure there's others. But where we are today is that like we hope like we'll get a biopsy of the patient. We can map the trajectory. Some patients will have ERB. Some people might have different immune signaling. Some might have other resistant traits. And as we get more and more patients, we'll learn more about these trajectories. And I'm hoping not every patient is completely unique, right? We kind of get a sense that there are some overlapping phenotypic trajectories across the patients. And then I'm hoping we can also build out to like not just first line, but like over longer periods of time, what happens to the trajectories. But at the end of the day, we, you know, at least right now have kind of the system where we have like patient A might be more sensitive, patient B and C might be more resistant. We can maybe use this to make as part of a model for predictions. Oh, yes.
Well, just curious, do they plan to do any ex vivo stuff where they take the cells and then they culture them and try from a particular patient and then they
Right.
train from? Right?
Yeah, it's part of my larger clinical trial. We don't, like for metastatic, this is the trial that I'm doing is in metastatic cancer patients. So we're getting both DNA, RNA, methylome, we're getting blood ctDNA, methylome, and we're getting all the clinical readouts. We're getting all that up front, and then longitudinally we'll get the circulating tumor DNA and RNA and methylome. We don't have enough sample after all that to do the growing the cells in culture.
Nothing left, I see.
Plus, you know, it's crazy because every time I take a patient cancer and I grow it in culture, I'm surprised at how delicate cancer is in vitro. You don't always get growth from a patient biopsy's cancer cells. They're very easy to kill, actually. And so sometimes I'll get a patient whose cancer grows gangbusters in
So the
culture.
microenvironment is what made a big
It's
difference.
really
right?
important, and they're, you know, they're not as hardy as we think they are. Yeah. Even in mice, ER-positive breast cancer doesn't grow in mice, you know, even in vivo. So they have different hormonal structure than us. So I changed my figure now. So like, yeah, we have that patient up front, right? Our end goal here is to say, okay, up front, we predict like this blue, you know, this light blue cell is a problem, right? So we treat up front with that combination therapy. You know, metastatic cancer will progress, right? So we'll have this, you know, clone pop up, but we'll identify it and treat it. This other patient might have a completely different trajectory, right? Where immune signaling is important. If we can tell that up front, we can start with different immune targeting therapies and then continue to kind of add on to that as we understand the biology. So this is just like in the clinical trial, we'll be tracking people through multiple lines of treatment. So it's important to think not just that one treatment, but like how do we build out models that look at different time points, different treatments, different trajectories. Yeah.
Can you just give, why don't you give at the beginning all the possible drugs you have?
Why can't we give all possible drugs?
Yeah, the black and the blue and all of them at the beginning all the time.
Well, okay, so I'll have two comments to that. First, there's a lot of side effects from drugs, so you got to give the right drugs, and you can't kill the patient from the drugs. Sometimes drugs do kill patients, so
Most of the time.
yeah, if you give too many drugs, or you know, you can have severe side effects, right? And even if you don't give too much, you can still die from drugs.
Even if you give them in smaller doses, like all.
Smaller dose might not be effective. You have to reach a concentration that's effective against cancer. So there's a dosing kind of component here. The second thing I want to say is we don't always know more drug is better. So if you think about the feline patients, the combination therapy, most of the patients that had that rapid progression, and you saw this in the endocrine signaling, rapidly turned down those resistance patients. They became endocrine resistant quickly. So they rewired more quickly when they had the combination therapy. More drug isn't always better. You just have to understand the dynamics of these relationships between drug and tumor response.
Right, timing can
Could be
have a huge
antagonistic.
effect.
Right timing have a huge effect, and
Right.
selective pressure. If you have too much selective pressure, then you kind of have this kind of burst of evolution where they can bypass normal restrictions.
Is it true to say that this is an optimistic picture where for each color dot you have a drug that kills it?
Oh, very optimistic. Yes.
Because usually if you have a resistant cell, there's not necessarily a drug that
Yeah, this is the problem we're having because in getting the drug is hard. So like if you think in a clinical trial where you want like a basket of drugs that you can just apply to any patient, you have to get agreements with every single pharma company to use a drug in this clinical trial, and they might not have drugs that target those resistant traits, or they
they
might
not even
not
exist, right? Is
Yeah,
that
they might not exist. The company might not want to use them with other drugs. You know, there's a lot of hurdles to getting this done well. But if we can provide the rationale, like if they can be more successful with their drugs, then they'll make the drug or they'll let us use the drug. So it's better than, so right now, right, you have a randomized clinical trial where you treat 2,000 patients with 2 drug regimens that are incrementally better. One might be incrementally better, but you don't personalize to the patients that will benefit from them, right? You just randomize, and then one population does slightly better, and then that drug gets approved, right? So we're trying a different approach here that might be more efficient for the drug developers as well as better for the patients That's my pitch for them.
Very convincing.
Thank you. I'm hoping it works, but I need the tools to prove that, right? Because if we don't bridge the math to the clinic, then we're kind of limited. So, yes.
So on that front, I know that you're collecting this awesome longitudinal data, and you said
1,700 patients.
How many?
1,700?
1,700 patients, which is awesome. That's not a lot by Google
No.
standards, but it's a lot
Yeah.
by cancer standards.
And it'll be longitudinal data.
And it's longitudinal, which is amazing. To the extent that, I know you guys have your own computational people, but boy, it's nice to be able to chew on interesting data. You know,
Yes.
how available is that going to be?
So I am a strong advocate for making data publicly available, especially if the taxpayer pays for it. So this is publicly known, so I'm still speaking from City of Hope. I'm just giving public information about the government trial. But as fast as possible. That means I'm not waiting for publication. I'm not waiting for anything. As soon as I can get the data to the people within months, then it will be out to the research community. The government owns the data. We want to share it with the community. So it's different than NIH funding. It's different than HTAN. It's different than everything you've heard, TCGA. This will be faster.
Great.
Yeah.
How frequently are you going to be sampling?
So for the clinical trial, upfront, it depends on, for that work, we're targeting 3 different patient populations, breast, lung, colon. They each have different time points, but I'll just give you a general kind of thing. Prior to each line of treatment, we'll have data. And for colon cancer, that'll be biopsies. For the other 2, it'll be biopsies upfront and then the blood-based data sequentially. So pretty much. Sometimes there's a biopsy at 3 weeks, so I'll just throw that out there, but that's only for colon cancer.
And the blood-based every week, every few days?
Depends again on the cancer type, but think every few months.
Oh, wait, the blood every few months.
Yes.
I see. And you do for circulating DNA, tumor DNA?
And methylome, and the circulating methylome from the DNA. Yes. And there's a pilot that has RNA.
And what kind of cancers are you going to look at?
Breast, colon, lung, metastatic.
Breast, colon, and what?
Lung. Non-small cell, ER positive, and triple negative, and colon. Yeah. So please use the data. Reach out to me if there's any hurdles or anything.
Just ask a cartoonish question about this cartoon that you
Okay.
have. So it sounds like a patient that has a strep throat and the flu at the same time, right? And basically what you're saying is if you treat them with an antibiotic, you end up sometimes with a patient that has the flu.
Okay.
Right.
Yeah.
But nonetheless, here it's still one disease, right? So how do we end up developing with this strange mixture of cells which are partly resistant, partly not resistant, and so forth?
Why do you think there's one disease?
It's evolution.
Yeah, it's evolutionary pressure on a heterogeneous population of cells.
There's a thing we give one name to that has lots and lots
No,
of
I understand, but what I'm saying within the same tumor, okay, I mean, that's a good answer, it's just evolution, but still, I mean.
But it's more than that because they also, like in metastatic cancer, for example, there's different sites which have different environments.
Spatial evolution.
Which, yeah, sorry. Spatial
Oh, but the
evolution.
short
There's a lot
answer
of-
is it's evolution.
It's
It's evolutionary
yeah.
pressures.
And they evolve quickly, so they're not like us, right? There's a lot of genetic and phenotypic heterogeneity across these populations, which allow them to respond to therapy quickly.
Can I,
Yeah.
just to make sure I learned something from this week, I also heard that there was a lot of, epistatic.
Epigenetic.
Epigenetic,
That's
right?
good, methylome.
Yeah, epigenetic.
Well, but that's what she's looking at the RNA. So how much, actually, but along those lines, I mean, with the kind of resolution that you have, you cannot answer the question that I would like to ask, which is how much of it is reversible epigenetic changes?
I don't know about the reversible,
You
but
don't
we're
know.
getting the methylome, which is epigenetic marks on the
Right.
DNA. And I estimate like 20% of the marks change from the methylome.
But I'm saying reversing back. but
Yeah,
you would
with
have to
treatment,
stop.
reverse it back. I
Right,
understand.
the question is, especially if you do metronomic therapy, which in your told there, you said that you are doing metronomic therapy. You said lower doses,
Right.
more spaced. So in that case, you might be able to see some reversibility or some.
Yeah, generally speaking, from the methylome data, there might be marks that are reversible with treatment. And the challenging math question there is you have a lot of signal to noise in that data, so you have to understand what the signal is. And so if you can do that, then you can answer that question.
Also, just as a side note, like sometimes you can get resolution that doesn't correspond to reversal, or reversal that doesn't correspond to resolution. Like these are phenotype, right? We don't totally understand what's causal.
No, but for example, the
necessarily,
stuff that I was showing in my slides, right, where in 12 hours in this cell culture, you had complete reversal to the MDR1 equilibrium distribution. 12 hours after application of the drug. If you took from the low, if you took it from the low MDR1, if you took from the high MDR1, 17 days, right? And you
I
had
see,
complete
I
reversal.
see. So
And
in
that's
that
experimental
context, I see.
results, right,
I
that
see.
we recapitulated in our model.
I see.
So that was
If
pretty
it'd
obvious.
be cool if there was like
But that was in vitro,
Yeah,
it
yeah,
was,
yeah.
you know.
It's very hard to see,
Yeah.
but that would be neat. Okay, I
Yeah,
understand.
that's what I'm asking. Can you do this?
You can try. Epigenetics typically takes longer than like transcriptional changes for us for some reason.
Oh, so when I say epigenetic, this is a discussion we had at the end of my talk, which was when I say epigenetic, I don't mean necessarily histone marks.
Okay.
I mean things that could be like change of expression through a
Okay.
feedback loop, you know, like SOS feedback
Yeah.
on the EGFR and things of that sort. Huh?
Elasticity.
Like, call it whatever you want. But it's not genetic. It's changes in equilibrium states of the
Oh, that is
bispecific
not
system
what-
or something.
So thank you for your questions. Happy to answer any more. This is the group. Ironically, Jason's not pictured here, but he did almost all the modeling. Aritro also did some of the work with the bioinformatics side, and Pat as well, Jen Fung, and then Pat and Rena are at the bench. So
Is Jason a postdoc with you, or is he?
he's a what is he? Visiting researcher professor. We've been working together for a long time. He's just fabulous. He has a background in ecology and mathematics. So then I moved him to translational genomics and and predictive modeling. So he's just jack of all trades, always knocks it out of the park. We're happy, to collaborate. So if it's something of interest, I only showed you some of the work today, but we have a lot more patient data and paired data with methylation and gene expression and a lot of cell-based data that's really tightly, that's beautiful. Much more pretty than the patient data, but the patient data.
The patient data is all City of Hope.
The patient data I showed today is Save Hope, all available on dbGaP. You can download it, use it. And the cell line data is always published with my papers, so all of it is there. And if you need something, just let us know. Yeah. Okay. Thank you.